So Dr. LeLorier is a professor at the University of Montreal in the departments of medicine and pharmacology. He's also the chief of the pharmacoepidemiology and pharmacoeconomy research unit at Hôtel Dieu at the CHUM. He's also an adjunct professor in the Department of Epidemiology and Biostatistics and Occupational Health here at McGill as well. He's been on several board member of several boards, so I've got a whole list over here, too long to mention now. He's published over 150 papers, he's published books and book chapters, and he's directed over 20 master's and PhD students. He's played a big role here with CNODES and is the site lead for the Quebec site, and he's a member of the steering committee. So we're looking forward to his presentation, which is very timely. And so the floor is yours, Dr. LeLorier.
Merci, Laurent. So, Laurent, just let me know if you don't hear me or something like that. So, thank you very much, Laurent, for your introduction. Hello, everybody. So today I'll be talking about the clinical development and deployment of COVID vaccines. Conflicts of interest. I received from Pfizer a fund for the development of clinical research of $80,000 per year. This I use as early career development for promising young clinical investigators, and it has been extremely useful so far. I do not personally benefit from these monies. Among the 25 masters and PhDs I have trained, 5 work full-time for Pfizer US, not Canada, excuse me. So objectives, to compare methods and procedures, to try to know what makes similar and differences between pharmacoepidemiology or pharmacovigilance and vaccinoepidemiology or vaccinovigilance, which is a new term I just made up. What I'm presenting today is the state of the knowledge as of today, 2 p.m. It is possible that by the time I am finished the presentation, many things might have changed. I've been giving this talk in several places and I have to practically redo it every week. So I'm trying to answer the following questions. Who should be vaccinated? With what? And then the main questions that we all ask ourselves is when are we going to return to normal life? So you have seen this guy many times. Some important numbers to know about the viruses. The first one is the rho naught, it's pronounced rho naught, which is a reproduction number. And that's the number of people who are infected by one patient. So as you can see, most numbers are around 2, 2.8 to 1.9. COVID is 2.2. Seasonal gripper is 1, so very low. I mean, the most infectious agent ever is measles, which has a R naught of 12. That is, one person infects 12 persons. And that is that as soon as herd immunity goes down, infections start reappearing. Oops. I can move to the next slide. Oops. Okay, and R naught is also very important because it determines how many people have to be immune to develop herd immunity. And there is an equation which I put here on Excel. And here you have the R naught. The initial R naught for COVID was around here, 2 point something. So that means it needed 60% vaccinated people or immune people to develop herd immunity. As the R naught goes up, you need more people immune to develop herd immunity. So right now, the UK variant is around 3.2. So that means that we will need about 70 people vaccinated to develop herd immunity. So R naught is an extremely, maybe one of the most important numbers concerning viruses. Case fatality rate, obviously very important. Ebola, which is the world record, is 50% case fatality. There's 50% of those infected die. COVID was 3.3. It probably has changed because more and more people are being infected, and these people do well, obviously. So the case fatality rate might have gone down as more younger people are being infected. Now, the key thing about this virus are mutations. And you have to realize that mutations are constant. It's been estimated that the COVID has mutated 40,000 times since day one, and it keeps going. And it is a stochastic process that is totally random. But at one point, if a mutation makes the virus more fit, then that virus takes over. What can make the virus more fit than the others? And we're talking about good old Darwin here. Well, it being more infectious. Obviously, a more infectious virus will be more fit and will take over. If vaccines are there, more resistant to immunity, not obviously everybody's nightmare, because the virus which is resistant to immunity will obviously take over. A point which is not clear, at least in my mind, is whether more serious infections or higher case fatality rates makes a virus more fit. Personally, I don't think so, but people disagree on this, because it's in the interest of the virus or the bug to keep the patient infectious for as long as possible. The reason tuberculosis and syphilis were so successful as bugs is because they kept people infectious for long periods of time. People would go on coughing and coughing and coughing for years and years and infecting people before they died. A virus that would kill people too fast would not be advantageous to the virus because it will make it less infectious. I will talk about that when we discuss the mysteries of Spanish flu. And the other thing to realize is that variants compete with each other. Right now we have 3 identified variants. They are competing with each other. Fortunately, the more infectious is also the less lethal, which is a UK variant. So with some hope that one will take over the other two, which are nastier. Now, this is the spike protein seen from above. That's where all the action takes place. The spike protein is what the virus is used to enter in the cell, and that's where the antibodies act. So they keep the spike protein from being able to penetrate the cell. Here you have 2 variants, and you can see that actually these are amino acids. So these are the sites, amino acid sites, which have become different and make the variant successful. Now, if you want to be a virology snob, then you don't talk about the UK variant, you talk about the B117, and you're even snobbier, then you talk about the 1.351. And that will sure way to make people hate your guts. Variants, this is a simulation of the speed with which a variant takes over. It's based on the UK variant. And you can see the simulation was done March 1 and is right on the spot, because today we're right where it was predicted by the curve. So it takes about 5 months for a variant of the UK type to take over the world. Now, variants become resistant to immunity, and that could be to previous infection or to vaccine. And that's obviously a huge problem. Now, it's important to realize this. If there is 0% immunity, if the patient is not immune at all, well, nothing happens because there is no selection that takes place. If there is 100% immunity, nothing happens because the virus doesn't live long enough to mutate. Okay? Now, if we go to 50% immunity, that's the most favorable condition for resistant variants. So by delaying the second dose, which is what we're doing now in Quebec and many other places, you put the patients longer in the sweet spot, that is in the 50% immunity. So I think it's a good decision because you decrease the number of cases. If you vaccinate everybody for 1 dose before you vaccinate the second dose, it's clear cut that you have less infections, and the more infections, the more variants. But we have to be aware of the fact that we're putting the whole population in the sweet spot, where if by random a variant develops, it has a bigger chance of being immune to the doses we're getting. It's interesting that nobody talks about that. I heard it mentioned at a conference where eminent virologists were present. Some of the guys, the Columbia virologists, said, well, do you realize, guys, that by delaying the second dose, we prolong the period during which we are likely to develop immunity? There was a silence of 15 seconds, and then they moved to something else. So it's obviously nothing we can do about it. I still think it's a good decision to delay the second dose, but there is a big price to pay which actually might end up in a total disaster. Immunity, in 2 words, immunity is composed of these 4 components. It's important to realize that the 4 components should work together, but they don't have the optimal for immunity to work. The component you mostly see is the antibodies, which are made by the memory cells. Now, the antibodies are easy to measure, so there are many papers on antibodies, but it's important to realize that antibodies are a proxy outcome. The clinical outcome is clinical disease, severity, and death. And they are not a very good proxy outcome. If they are high, the vaccine has high antibodies, that's good. If they are low, it's not necessarily bad. For instance, hepatitis vaccine is very successful in spite of the fact that it produces very few antibodies. So it's a whole system working together that will produce what we call immunity memory, which makes that when you are exposed for the second time, you have a robust response and you get rid of the bug. Now, vaccines, the idea of vaccine is to mimic infection, and it's a fascinating story, but I don't have time to talk about it. The idea is to produce immunologic memory. So far, we have 2 types of vaccines. The vector vaccines, where is the immunity-producing protein is stuck into another virus, which is a nice virus. And this is the case for AstraZeneca and Johnson & Johnson. It's important to know this, we'll see later why. While the mRNA vaccine, which is really an amazing thing, what you give the cell is instructions to make the vaccine. So it's like you go to IKEA and you come back with plants, but you have to find your materials yourself. Many people thought that vaccine would never work. It had never been used in humans. And now I think we're damn happy we have it because it's going to be a game changer. Development. So let's compare drugs and vaccines. In drugs, we have a phase 1 and a phase 2, as you all know. In vaccines, we fuse phase 1 and 2 in what is called the early phase. That is, we measure the tolerability and to efficacy to some degree. So phase 1 and 2 are fused. I'll give you an example of a vaccine, which is the mRNA SARS-CoV-2, which was published. This is a phase 1/2 of Moderna. And you have the side effects during the first 7 days. And these are the classical side effects you get when they inject something in your body. Important thing is the side effects are more important at the second dose, as you see in yellow, than in the first dose. And actually, this is important because some people might chicken out of the second dose because they got nasty side effects of the first dose. I have a colleague who had a huge reaction to the first dose, and she's really scared about getting the second dose. So we're trying to convince her to go ahead and do it. And then efficacy, since it's a mix 1 and 2, what they do is they measure the antibodies. What you have here is the 3 doses. And this is the antibodies before the injection. And these are the antibodies after the injection. For dose 1, dose 2, the same thing, before the injection, after the injection, before the injection, after the injection. So a very robust response antibody-wise. And here what you have is extraneous data. That is the levels of antibodies you would have expected if the patient had had the disease. So the vaccine is producing antibody levels extremely similar to those obtained with the disease, which is obviously very, very nice. Now we go to phase 3. And here we have the BNT162b2. I mean, you really want to do a virology snob, then you mention the vaccines by their code number. And if people look at you with round eyes, well, you sort of despise them and inform them that actually this is a Pfizer vaccine. Okay, so that's the phase 3 of Pfizer. They randomized 48,000 patients. The plan was to randomize 15,000 per group. Now, when they look at the data, they did not have enough high-risk patients so FDA told them you have to go ahead and do more patients, not patients, excuse me, individuals before you get the data, because the FDA wants the company to have, if a risk factor is present in the population, it has to be twice as present in the sample which gets a phase 3. So they have to increase to 18,000 per group so they would get enough high-risk patients. Now, this is very interesting. A few words of PPPE, which is one of my favorite hobbies. As you probably know, there is a landmark paper on PPPEs, which is pharmacologically pertinent period of effect, which was published by the dream team of pharmacoepidemiology composed of Melanie Suissa and myself. It's useful to don't get confused with all these Suissa publishing papers left and right. When people in pharmacoepidemiology, when they talk about Dr. Suissa, they mean Melanie. If they want to talk about Sami, they talk about the other Suissa. Okay, so when should you look at side effects? Well, side effects are divided in two groups. The first group is the first 7 days after the injection, because these are the classical side effects you expect to get. You get the injected foreign body, so irritation on the injection side, some fever, some fatigue, and so on. And then there is another set of side effects, another PPPE, which is 7 days after the first 7 days, which are meaningful for other reasons. And now, very interesting, in clinical efficacy study, that is phase 3 study, the efficacy they start measuring efficacy 14 days after the first injection. So an infection occurs between 0 and 14 days, it doesn't go into the statistics. The idea, of course, is that it takes 14 days to develop immunity. And this being an efficacy trial, they obviously want to put the drug in the best possible situation. This is not what happens in effectiveness studies. So that's, I think, a very interesting application of the PPPE principle. Now let's talk about effectiveness. Okay, the first effectiveness measure is geographic statistics. Those are the ones you see in the newspapers, on TV every day. But all these statistics have problems. Number of new cases. Well, I mean, the number of new cases will in part depend on the number of tests. And even Donald Trump, with his fine, acute, analytical mind, quickly realized that if you don't want to see new cases, you should do less tests. So a number of tests can vary according to the facilities of the population. The percent positive tests, if people are very scared, they'll be tested more often if they're not scared. I have a colleague who's been tested negative 12 times. Number of hospitalizations can be biased because the hospitals become full. That's becoming right now, unfortunately. Admissions to intensive care is also limited by the number of available beds. A number of deaths by COVID is not that easy because the definition changes and it's not the same in all countries. The state of New York changed their definition and then their statistics change. And you have to distinguish between death by COVID and death with COVID. And a terminal cancer patient who has COVID and died, is that a death by COVID or is that a death with COVID? So it's not as simple and straightforward statistics as you might think. And I would really like people to have the denominators. The object of these statistics is to compare countries with each other and countries in time. And this cannot be done when you don't have denominators. I don't have all these numbers in per 100,000 inhabitants. But obviously, I can work them out myself, but it would be nice to present it that way because it makes the numbers more meaningful. And finally, my favorite statistic is almost never mentioned, which is excess mortality. How many extra people died this day, today, or this week compared to a similar week pre-COVID? Well, it is not part of the regular statistic, but you can, thanks to Google, work it out. This is what I found. 219 pre-COVID, Quebec 67,000, post-COVID 74,000. So an excess mortality of almost 7,000. So I think that tells me a lot. And I'll know the epidemic is over when we go back to 77,000. Now, when looking at this, I ran into this fascinating graph that I'd like to share with you. These are Quebec data. Here you have in the y-axis the number of patients, number of deaths or whatever, and 100 is the baseline. So 0, excuse me, is the baseline. So it's the average of the 4 previous years. So this is the expected number here. So 0, no difference. In blue, you have the total deaths, and you see they are hovering around 0. And then all of a sudden here, COVID appears, and you have a huge jump in total deaths, the blue one. And then the first wave goes away, and we start confining people. It goes down, and then the second wave comes in, and the total deaths go up. But if you look at the COVID deaths, you have an excess death of COVID compared to total deaths. Now, if nothing else was happening, the additional number of deaths by COVID should be equal to the additional number of deaths, all causes. This is not what happened. We have almost here at the epidemic time, 150 excess deaths with COVID compared to the general population. Then it gets the same. And then when the second wave comes in, the same thing happens again. You have more deaths by COVID than could be accounted in the total population. This tells me that something else is taking place that is decreasing the number of deaths when we get confined. And what could that be? Well, we thought about car accidents. There are not enough car accidents. Surgery is not enough surgery. What people die of? They die of cancer and cardiovascular disease. Cancer takes a long time to develop. So we said, and some experts help us in that, it must be cardiovascular disease. So what you have here, so an article in the New York Times, where have all the heart attacks gone? Except for treating COVID-19, many hospitals seem to be eerily quiet. That's not the case of my hospital. And here you have a NASA data on nitrogen dioxide, which is the main pollutant. And this is the east coast of the United States. Unfortunately, we don't get to Montreal. And you can see that there is a lot of nitrous oxide in the atmosphere in regular times. And then you go to a confinement period, and then you see that by confinement, the pollution goes down dramatically, but really dramatically. And obviously, this is in part due to much less car driving around. So confining produces a clear-cut decrease in NO2 emissions. This is very nice too. This is a time graph. In yellow, you have Wuhan, the original province. So this is the pollution in Wuhan. Confinement appears and the pollution drops down. In blue, you have Madrid. This is pollution in Madrid. Madrid lanes get confined, pollution goes down. And here in green, you have New York. Confinement comes in, pollution goes down. So there is a very clear cause-effect relationship between confinement and pollution. And there is also a very nice paper by Chinese investigators that show that for every 10 micrograms per cubic meter increase in NO2, the number of cardiovascular deaths, cerebrovascular death, and ischemic heart disease death go up significantly. So it would appear that that's a hypothesis. We were working very hard on it, and I have some very good people on that, among others, François Reeds, who wrote 2 books on that. But it would appear that the less cardiovascular deaths are really due to less pollution by confinement, and that's explains the discrepancy of the slide I just showed you. Okay, Israel and COVID-19, beautiful story. So Israel has a population of about 9 million, and the Prime Minister of Israel one morning said Israel will share with Pfizer and with the entire world the statistical data that will develop strategies for defeating the coronavirus. Now, Israel has a universal and obligatory healthcare systems. They have 4 HMOs with very, very good data. Now, what are the details of the quid pro quo? Who gave what to whom? Well, they have never been published, so we don't know what was the deal. But apparently, Israel paid more for the vaccine than the other countries. Now, when I found that out, I was really very sad, because I said to myself, I mean, if the finance minister of Israel can negotiate a good price, I mean, Trudeau, that poor little twit, he doesn't have a chance. He must be paying the double. Anyway, the deal was that Israel will give the data and Pfizer will give the drug faster, the vaccine faster than Israel could distribute. So the limiting factor will not be availability, but distribution, which I think is a fantastic deal. We could have done the same thing in Canada, but obviously it's not possible. So there is another paper, but it's a lousy paper. This is the really good paper. So as you can see, the BNT162b2 vaccine, which I remind you that it is a Pfizer vaccine. Now, if you want to be a snob of virology, you don't have to memorize these numbers. You can make them up on the fly, and nobody will find out that you're just inventing the whole story. Well, okay. So the study is all people vaccinated between December 20 and February 21. So it's really the very first of a beautiful series of studies I can only wait for. The methodology, they match one-on-one vaccinated people with non-vaccinated people. So the people who got vaccinated were the exposed, and the non-vaccinated were the controls. Now, obviously, this is an observational study. It's an observational study about the self-selected population. That is, people decided themselves they wanted to be vaccinated or not. And whenever the exposure is self-selected, it brings in tons of confoundants. Same thing with exercise and diet and so on. Because there were two factors here: probability of being vaccinated. The guys who decide to be vaccinated are not the same in many factors as the non-vaccinated, and the probability of infection is not the same in vaccinated and non-vaccinated. So there is a huge potential confounding here, which is almost infinite and actually quite fascinating, because among other things, it could be that those who decide to be vaccinated are also more responsible people and they take more precautions. And you can invent confounders like that all day. So how did they deal with it? The outcomes: symptomatic COVID, COVID with acute respiratory syndrome, COVID with hospitalization, death of all cause. So obviously all the right outcomes, these guys know what they're doing. Results. This is amazing. They have two groups of 596,000 individuals, which have been matched for 12 clinical factors, which are factors which change the probability of getting the vaccine or getting the disease. Now they have the usual first table is, characteristic of cases and controls. And you look at that table and it's amazing. You would think you're looking at a randomized clinical trial, because all the subjects are very, very similar according to all clinical and demographic parameters. It's just when you get to older people, they had some difficulty sometimes in finding the right control. So things are forced there. But otherwise, I think this is a landmark epidemiological study that we all wish we had the data to do it ourselves. The results. Well, very quickly, classical Kaplan-Meier curves, unvaccinated in red, vaccinated in blue. As you can see, beautifully, the curves separate at 14 days, which is exactly when the immunity starts. This is after the first dose. So this is cumulative incidence. This is the documented SARS, that is SARS with the lab test. This is symptomatic COVID, not SARS-CoV, excuse me. Same thing. This is hospitalization for COVID, same thing. This is severe COVID, same thing. This is death from COVID, same thing. And something which is really very different from what we get in pharmacoepi is that the efficacy and the effectiveness were exactly the same. So the drug worked as well in the field as I work in the phase 3 trial. Actually, this is not that surprising, because, there is no problem in the administration of the drug. Unless the person is very stupid, you cannot misadminister an intramuscular injection. So the drug is always well given, and we know exactly who got the drug, the vaccine. So one of the reasons efficacy and effectiveness are different in pharmacoepi is that on the field, the drug is often given to the wrong persons, the wrong doses, the wrong interactions, and so on. Safety. Well, before we get into safety, we have to talk about something we don't often deal with, at least us, is cluster epidemiology. Now, the classical cluster is a geographic cluster. Classical case of a geographic cluster, 4 kids with leukemia in a town of 10,000 in the same year. So that was a cluster of cases of leukemia. I must say that my mentors in pharmacoepidemiology, which I won't mention because I want to embarrass them, the 3 of them told me the same thing: don't touch it. Whatever you do, don't get pulled into a cluster study, because it's hell, it's very difficult epidemiology, and it's very emotional. Because obviously, you can work out the incidence of leukemia, and you can see this is extremely rare, it cannot happen by chance, except that you're not talking about one town. We're talking about 100,000 towns of 10,000 people in a Western drug board, and that's how you should look at it. So then anywhere in those towns at some time, a rare disease can be appearing for kids. So those are geographical clusters. In vaccine, we did have something else with these time clusters. That is a certain number of cases in a certain period of time, which corresponds to the period of time which immediately follows the vaccination. And these are the clusters that were identified in the phase 3 trial. Between Pfizer and Moderna, there were 9 cases of Bell's paralysis. This is facial paralysis. Those who are old enough to remember Jean Chrétien, well, you know what facial paralysis is. There were 9 cases between Pfizer and Moderna together, and 1 case with the controls. With Astra, you had 2 cases of transverse myelitis. Transverse myelitis is a horrible disease where you become paralyzed, and it's really something which is quite horrible. And there were 2 cases in the vaccine group and 0 cases in the placebo group. So these are the signals that were identified at the end of the phase 3 trial. Now, why is it such a big challenge? Well, first, the advantage. In phase 4 trials or studies of vaccines, we have a huge advantage over drugs. They have very, very good denominator, particularly when it's a vaccine like COVID. It's not true for other vaccines. But in the case of COVID, everybody, every country, every health state knows exactly who was vaccinated with what and for how much. So that's very much better than we know with drugs, because drugs, we don't know. We know how many drugs were sold, but we don't know who got it and at what dose and whatnot. The denominators are fuzzy because they are subject to detection bias. Obviously, people who have been vaccinated, everybody is watching them very closely, see if something goes wrong. And then you have publicity bias, that if a given so-called side effect is identified, then it gets into the newspapers, and then physicians are looking for that side effect in their patient. And then you have a publicity bias, which is usually also in drug epidemiology. It's interesting that the best comparator in the world of drug with vaccine is oral contraceptives. Because in vaccine, as in oral contraceptives, you have an intervention in many people in good health, and you expect them to stay in good health, and so the tolerance for side effects is very low. And then what we have, obviously, is we have a temporal association. The person got vaccinated, and 10 days after she developed whatever. And of course, the big question is this temporal association, causal association. I think it's important to know that, in my opinion, the human mind, human brain is wired to assume that something is temporal, is causal. And there are many reasons for that. But the first reflex when we see a temporal association is to assume that it's causal, It's the reflex of the public and the journalist, and it's our work, our job to bring them back to reality. So what do we have? Well, you have the expected, that is the annual incidence of the disease, how many cases were expected, given the number of people who, given the incidence of the disease, and how many were observed. And when they observe, the big question is when? What is the pertinent pharmacological period of effect? Is it 1, 2, or 3 months, or a year? And you have to choose a time window, and the time window you decide to choose can give very different results. Usually, the vaccine people use a 2 month PPPE. Now, see, let's look at this. AstraZeneca phase 3, 2 cases of transverse myelitis. The annual incidence of transverse myelitis, hopefully it's a very rare disease, is 1 over 220,000. So what is the probability of having 2 cases in 15,000 people in 2 months? Well, obviously it's very, very low. It's 1 in over 20,000. But that's not the answer. The answer is, if we have 100, this is a simplified modelization, if we have 100 different independent diseases, all of them with a yearly incidence of 1 in 220,000, the probability of 2 cases of any one of these diseases in 15,000 patients is 30%. So it's not the same. And the real number is 30%. If you go by the 1 in 20,000, then you will decide that this is prohibitive and that the drug should not be given. So the signals generated by phase 3 mRNA vaccines, as I said, Pfizer and Moderna, facial paralysis. When we got to phase 4, the signal did not reappear, it disappeared. There was no higher cases of facial paralysis, and the same thing for triphase myelitis. So the 2 signals generated by phase 3 were not confirmed in phase 4. And that shows that the signals generated for phase 3 are really a hypothesis which needs independent confirmation. So where are we now? Well, there have been 18 cases of a very rare case of thrombosis post-AstraZeneca in the UK and Europe, which have been 20 million vaccinated. This is a hypothesis. This doesn't prove anything. Then the Germans just published 3-4 days ago 12 new cases which are independent. They were not part of the initial 18 cases. And in their publication, they already jumped the gun and decided it was causal because they invented a new syndrome, vaccine-induced thrombotic thrombopenia. So this is independent confirmation. So the AK European were the hypothesis, and the German case is dependent confirmation. And as a further nail in the coffin, Janssen reported 2 cases. And interestingly, as I said before, Janssen has a mechanism of action similar to AstraZeneca, which is a common vector. So on top of that, we have biological plausibility. So as far as I'm concerned, there is clear-cut causality with very rare but very ugly disease. So what do you do with that? Well, first, are there any alternatives? Yes, there are other vaccines. Second, what's the risk benefit? Well, I mean, that requires a specific study or a risk benefit study, which is actually an economic study with quality of life and so on. It's a Markov study. I haven't seen it. I haven't done it myself, but the authority tells us that the risk benefit is favorable. I would like to see how they work it out. Second possible alternatives is identify subgroups. This has been done. And they noticed is that all the cases were in younger people. So they decided to use the vaccine only in people over 55, because there are no cases over 55. And then Quebec yesterday just brought it down to over 45. And then the other option is sit for 3 months and wait for the mRNA vaccine. In the US, you can choose your vaccine. They have much vaccine. You have here in Quebec, you cannot choose your vaccine. You get what is available, but you could potentially wait if you're scared of the AstraZeneca Johnson & Johnson vaccine. Wait until the mRNA vaccines become available for everybody. So there are options, there are alternatives. The problem is in part psychological. That, as you know, people are scared of vaccines, and that's reasonable. I'm not talking about vaccinophobic people or militant people, but some people are what we call vaccine hesitants. And we need these people to get vaccinated if we want to reach herd immunity. And all these data they have difficulty understanding. What they tell you is, how do I know it's not going to happen with Pfizer and Moderna? Now, let me tell you one thing. The safety of Pfizer and Moderna vaccine is amazing. We haven't had one single signal in 100 million vaccinated people. I mean, any signal worth working on. Now, obviously, our Israeli friends are going to give us all the data with all the nominators and so on. And I expect to see a lot of beautiful papers from this guy who will answer many, many questions. Predictions. So what are my predictions? First, it is difficult to make predictions, especially about the future. This comes from Mark Twain. I see three scenarios. This is very personal. I see three scenarios. One is the Spanish flu scenario. The other one is a multi-vaccine scenario. And the third is confinement ad vitam aeternam. Spanish flu. Fascinating story, but I don't have time to tell you. But by going to Norway and Alaska and looking at the graves of people who have been buried below the permafrost, so the virus was still intact. So they went in and made holes and got these people and froze the samples and sent them to the NIH. And they also used soldiers' autopsy, which were at the pathology department of Walter Reed. And with this task, a lot of very competent people were able to work out the sequence of the Spanish flu virus. It's really a detective story. I wish you could read it. I advise you to read it. So they found out exactly what was the sequence of the virus. It took lots of work for many people. And then they came out to the conclusion that the seasonal flu, the good old seasonal flu, is actually a drift mutation from the Spanish flu. So slowly the Spanish flu mutated to become the seasonal flu, which is actually a very successful virus because it's going to be with us forever. It makes people symptomatic without being very sick, and it has an amazing ability to mutate. So we have to change the vaccine every year. So this is, from the point of view of the viruses, that's a smart move because that virus will be there forever. Now, if this happens with Coxib, well, it will not be that bad because after all, we can live with a yearly vaccination as we do with the Spanish flu. But nobody knows if this is what's going to happen as immunity starts to develop. Second scenario, there is a limited number of resistant variants. So variants through the world tend to converge. So the Pfizer people, not only they discovered the mRNA vaccine and Moderna too, but they are working on future vaccines. They tell us that they can develop a new vaccine for if the virus becomes resistant to the present Pfizer vaccine, they can develop a new one in 2 months. Let's say vaccine A was effective, okay, then comes B, comes mutant B, they can make a vaccine which is effective against A and B. And if mutant C comes in, they can make a vaccine which is effective against A, B, and C. Now, the fact that they can develop in 2 months, you still have to consider it takes at least a year to deploy it in the population. So they can fabricate the vaccine efficacious against several variants. And I believe them because those guys know what they're doing. So one possible solution or future of the problem would be that we have 2, 3, or 4 vaccines And then hopefully the virus has played all its cards. It doesn't have any way to fabricating any new vaccine resistance mutants. And then we have a vaccine that will work for all the viruses. Now, this is a nice part of the story. Less exciting is the fact that each one of these cycle, that this new vaccine deployment, new virus, is a 2-year cycle. So if their virus has 4 cards, 4 possible mutants that are resistant, well, we're talking about 8 years. And finally, the bad case scenario, confinement ad vitam aeternam. And as Woody Allen says, eternity lasts a long time, particularly at the end. And I cannot finish without mentioning the other American philosophers. The United States has produced 3 philosophers in all its history. This is Mark Twain, Woody Allen, and Yogi Berra, who famously said, "It's not over until the fat lady sings." Thank you for your attention.
Thank you very much, Dr. LeLorier, for your very informative and I would say also entertaining presentation. So the floor is open for questions.
Jacques, thank you very much. This is the other Dr. Suissa.
Yeah, I recognize you.
You mentioned the Israeli observational study. What would it take? What would it have taken for Canada to conduct this kind of a study?
Well, actually, I was talking with my Pfizer's ex-students. Some of them have done very well. I talked to them informally and I suggested a study, and they say, we're game. Well, first, the study cannot be done in a province because the vaccine is federal. So it would have to be a CNODES study, okay? And it would have been with federal approval and so on. But we have certainly the capability to do it if they provide us with a list of vaccinated people. There's nothing for the people we have to cross-link the vaccine with our databases. I didn't get to first base. When I started talking to people at the cabinet, at the minister, cabinet, prime minister, or some people I know, they practically got hysterical, accused me of selling the country to the pharmaceutical industry, and it just was a no-go. So we won't even talk to the prime minister about it. Now, in Israel, it wasn't easy. You know, all the confidentiality people and all those people raised hell, okay? But I think they rightly decided that it was worthwhile. First, themselves, Israel has the highest rate of vaccination of any country in the world. Okay, they are vaccinated very, very fast, except that they have some people who obviously refuse to be vaccinated. The prime minister said, well, I'm going to put in the newspaper the names of the guys who don't want to be vaccinated. So they raised hell. So I think he put in the newspaper the names of the guys who did get vaccinated. So you can find out if your neighbor was vaccinated or not by checking him out. But we could do it. We could do it. It's just people don't understand that collaborating with pharmaceutical industry is collaborating with the devil. We can say many bad things about the industry, but we won't have a vaccine without those guys. Now, those vaccines were developed by Pfizer using data which was developed by basic science scientists over many years. Okay? So I hope that answers your question.
No, the study that was conducted in Israel was studied in one of the HMOs. Israel has 4 or 5 insurance companies. They're all public. It's public funded, but they compete with each other. Yeah. And in a way, one of the ways to compete with each other is that they have each one has direct access to the data of their patients so that they can improve the management of the of the patient, they can improve health, they can identify early signals. But they have a bit like, I think Ontario, a couple of provinces in Canada have direct access, you know, they have scientists who have direct access to the data and can do this ongoing surveillance. In Canada and in Quebec, I can talk about Quebec in particular, it's almost impossible to have access to these data instantly so that questions such as these can be looked at. Side effects, for example, of the vaccines is an issue that you can look at when you have a very large population. You can look at instantly who's been vaccinated and who has these side effects. So somewhere there are several missing links to be able to do what was done in Israel.
Yeah.
And somewhere, are we going to get there? Are we going to be able to protect the health of the public with data that we have, but that we cannot have access to?
Well, I would hope so, but I'm not very optimistic. I mean, Israel, they work out a fantastic deal because they have all the vaccine they want. Everybody's getting vaccinated very, very fast. And what Pfizer gets is access to the data. Pfizer cannot publish.
They don't get access to the data. The data is accessed by the research team of Clalit, and they show them the results, but they do not have access to the data.
No, but they have what is almost access to the data. And it says the data are not sent to New York, okay? But they ask, if something goes wrong, they'll know it right away.
Exactly. Okay. Exactly.
That's what I talk about. That's what Pfizer gets about it.
Right.
That if there is a signal, they'll know it before everybody else, and then they can plan a strategy to respond to that signal.
And that is independent of Pfizer, because I think the public health investigators in Israel want to know as well immediately if something is wrong with whatever they are giving their population. So there is also that political will, independently of whether Pfizer gave them money or gave them a break or gave them a discount, 2 for 1 or whatever. Independently, I think the public health philosophy is that we want to know right away what we're doing to our population. And if we are harming them, we want to know and ajuster le tir immediately. And for that, you need instantaneous data.
Yeah, but the team that published, because there's another team which did a lousy study with another database, which I won't even mention. And it's a lousy study. But these guys know what they're doing. And they are exploring side effects day after day. And they know how to interpret the signal. They're not going hysterical because somebody develops a bad disease. There is one landmark point, Sammy. The moment a vaccinated individual gets hospitalized, we know we're in trouble. Because the vaccine does 100% protection against hospitalization. So as you saw in the data I showed you, they had some data, some infections, but not hospitalizations due to COVID. When hospitalizations due to COVID start in vaccinated people, we will strongly suspect that the variant resistant to the Pfizer vaccine is showing its head. It's going to show up. I mean, and I think delaying the second dose is making things more favorable.
Right. And this is something that could be studied with real world data, especially in a place like Quebec, where we have delayed the second dose for a very large proportion of the population. I'm going to stop asking questions here and I'll let others. Thanks, Jacques.
Thank you. Any additional questions? I guess it was clear. I guess I'll say one comment. I mean, we do see, I think all of us have been confronted with a lot of misinformation about vaccines. Certainly, I get a lot of these messages through WhatsApp, sometimes from family members or others that I know. So where do we go from there in trying, at least from a pharmacoepidemiological standpoint, to try to counter some of those claims or investigate some of those claims? So we're talking about the Israeli studies. There's one study that actually came out that looked at the impact of the Pfizer vaccine on fertility. So really, the whole basis for that, it seems, was not founded on any real hypothesis, but it's a study that was conducted to really counter those claims. So where do we go from this, and how do we choose those studies that need to be investigated, either because we really have a genuine concern about a safety issue of a vaccine, and the other side, which are those trying to counter some of those claims that are made that don't make any biological sense, but we feel that we need to investigate to bring more reassurance in the public. So I think maybe it's more of a question of how do we prioritize that kind of research, assuming we have the data?
Well, I mean, the guys who are going to do all the work and provide all the information are the Israelis, because they have numerators, they have denominators, they have a lot of vaccinated people. 500,000 people have been vaccinated in February. Okay, that's a lot, and they are moving very fast and have a lower incidence of the disease than any country in the world. Okay, and they just relaxed the confinement. Now beginning to relax the confinement. Now, if you don't have those data, as I said, cluster epidemiology has to be understood. Cluster epidemiology has two phases: a signal and an independent confirmation. And I think we have to educate that, because the signal gets the publicity, and the independent confirmation becomes seen by the public as scientists talking to other scientists. But because ideally we would like to reach herd immunity. But as I said, herd immunity with the present data, with the present bug, requires 70% immune. 40% have said they don't want to be vaccinated. So what can you do? And what do you do if you are vaccinated? But eventually, like most of my colleagues, I got my first dose. By the way, I waited until 60 million had gotten the Pfizer vaccine before getting my first dose. And so I got my first dose. I'm in between. Sometime next month, I get my second dose. I still have to take the same precautions. Okay. And after the second dose, I still have to take the same precautions. So eventually, what we want is, when are we going back to normal life? Okay. Because being vaccinated gives me some sense of security. If I catch the bug, it won't be too bad. I'm less likely to catch it, but I still have to take the same precautions. I cannot go back to normal life. And that's what people would like to. And we're paying big consequences, which are getting bigger and bigger. I mean, a lot of kids are not being exposed to normal schooling, among other things. We don't know what this is going to do to this generation. So it's a very complex, as you all know, problem. And unfortunately, I'm a pessimist. And as I said, the advantage of being a pessimist is two. The first one, we pessimists are usually right. And the second one, if I'm wrong, that means that something good has happened. So let's hope I'm wrong.
Well, thank you. So on that note, let's hope for the best. And we, as part of, so on behalf of CNODES, I'd like to thank you for your great presentation, very timely. And I thank everybody for attending. So on to the next seminar next month. Thank you, everyone.
Bye, everybody. Thank you for coming.